Okay, so it's my pleasure to introduce our first speaker today, Allison Wise. She graduated from Texas Lutheran University in May of 2021 with a degree in molecular biology. She spent the summers of 2019 and 2020 working for Mississippi State University at the Thad Cochran National Warm Water Aquaculture Center under the supervision of Dr. Matt Griffin. She recently started a master's program at Auburn University with Dr. Tim Bruce. So thank you very much, Allison, for joining us, and you can take it away.
Okay. Yeah, so like you mentioned, this was my undergrad research that I did 2 summers ago, my junior year. And we focused on genetic characterization of the Vibrio species isolated from commercially cultured fish. Okay. So the catfish industry is the largest aquaculture industry within the United States, and the Mississippi Delta relies heavily on the industry's revenue, producing nearly $340 million per year in the United States. And the 2 images below there are just to show you how large the ponds actually can be. And so disease within the catfish industry poses a threat to production, resulting not only in mass mortality, but treatment expenditures and lost feed days as well. And recently, farmers have reported spontaneous mortality within their hatcheries. And due to outbreaks being phenotypically and genetically consistent with Vibrio, the suspected causative agent has been determined to be Vibrio species. So Vibrio is a gram-negative rod-shaped bacteria, and clinical signs include anorexia, lethargia, abdominal distension, along with hemorrhaging, lesions, and gill necrosis. And this first image here was taken by Dr. Griffin, and these are fingerling fish, and you can see the distension in the bellies. And I'd also like to note that these were the fish that are also used to do the histology. And then to your right here are the what the infection could look like in carp, and you can see the hemorrhaging underneath the fish along with the gill necrosis. So there are several environmental factors that can facilitate infections within hatcheries. The most stressful being overcrowding and high temperatures can contribute to facilitating the infections. So the implications of what the Vibrio species infections could cause. At present, reports of Vibrio related mortality within the hatchery are only within the hatcheries, and Vibriosis can be a potential emergent disease which can cause mortality in commercial production systems, and characterizing this pathogen could aid in future diagnosis if it does make its way into the ponds. So in order to assess the risk posed to both human and fish health, the Vibrio species must be characterized and differentiated from other subspecies. So for culturing the isolates, they were provided from cooperating laboratories across North America from a number of different hosts. And we collected 66 isolates, and they were grown in the TCBS agar and incubated, and then the presence of color and growth was recorded. So for my, I really wanted to characterize them to see which primer sets would be best to differentiate the different species. So I started with a 16S initial molecular screening, and then a gyrase B would be used to confirm the species identity. And Pyrena H, Rec A, and RPOA were used as my differentiating primers, and they're also used to build a concatenated phylogenetic tree. I also ran a PCR screen for the Vibrio collar toxin, which I'll go more into detail in a second. And a RET PCR and MLSA analysis was also done to build a phylogenetic tree. And a disk diffusion assay was run to see antibiotic sensitivity against the isolates, and a BBL crystal kit analysis was also run as another biochemical way to identify the isolates. So for my TCBS agar results, 55 isolates were positive for yellow colonies, as you can see in the right photo, and 2 isolates were negative for growth on the TCBS agar, even from initially starting them on blood agar plates and then trying to transfer them to the TCBS agar. They were still unsuccessful in growing on it. And then 6 isolates presented this greenish blue colony, which is on the left. And so I want to point out that this yellow colony growth is consistent for the phenotypic characteristics for the Vibrio species. And these results correlate directly with my sequencing results. So the 2 isolates that were negative for ever growing on the TCBS agar were actually an Aeromonas hydrophila and Pseudomonas strain. And then the 6 isolates that presented this bluish green coloring that you see to the left were Vibrio vulnificus. So like I mentioned, I used a 16S, Gyrase B, Prina H, Rec A, and RPOA primers for sequencing. And they were prepped and sequenced with the following PCR protocol. And then the amplicons were gel excised and purified and then sequenced commercially. So this is just the first 24. As I mentioned, there are 66, but I would like to point out a couple things that the Gyrase B primer highlighted in red was the most successful, producing the most consistent amplification and the highest sequencing yields, while the 2 primer sets highlighted in blue, the Prina H and RPOA, were actually not as successful in having consistent results. But after Dr. Griffin redesigned them, they did have more consistent sequencing yields or higher sequencing yields, giving us more consistent results, but still not to the rate that Gyrase B was. And then the species identified through sequencing, there were multiple strains of Vibrio cholera, along with Vibrio vulnificus, Vibrio mimicus, Vibrio meticis, and Vibrio harviae. So for the cholera toxin screen, only O1 and O139 serogroups can cause severe symptoms in humans. So in order to assess the risk posed to the hatchery workers, this screen was designed in order to determine which strains have the potential to create the toxin by looking for presence of the CTX-AB gene. So a SOD-B primer would confirm the Vibrio species, and then a CTX primer was designed to confirm the toxigenic strains that contain the CTX-AB genes. A temperature gradient was run to find the optimal annealing temperature, and then the following PCR protocol was run. So to your left is my CTX gradient. I wanted to see the highest temperature at which both bands for the primer sets were present. And it was decided that E, which is coincides with 56 degrees Celsius, would be the temperature for annealing. And initially, I ran all 66 isolates against the screen, but the results were extremely muddy and there was a lot of non-random banding. And this could be because the SOD-B primer was made specifically for Vibrio cholera. So then I excluded all non-Vibrio cholera isolates, and then the results for that is what you see to your right. And as you can see, the banding is much more concise and more reliable than what we had done initially. And at first, I thought isolate 24 that you can see highlighted in red was potentially a positive band, but after further investigation, it's actually a little bit higher. It's bigger than what the positive band should be. And so that could also be from random banding. So for my rep PCR, I used 3 different primer sets, GT-G5, ERIC 1 and 2, and ERIC 2. And then we used the following PCR protocol. And the PCR products are run through a 2% TBE gel to avoid warping and ran against a 50 base pair hyperladder. So these are my results for that. These are the gel images for all 3 primer sets. And highlighted in blue to your left are the ornamental Vibrio cholera isolates. And as you can see, the fingerprinting is extremely similar between the isolates and between the primer sets. And in yellow, the isolates were very noisy. Now these are the mimicus isolates with varying hosts. You can see the fingerprinting, they're not similar at all. And then in red are the Vibrio vulnificus, and they have identical fingerprinting for all 3 primer sets as well. And highlighted in green are the catfish isolates, which you can see have more variation in fingerprinting than the blue, which is the ornamental Vibrio cholera. And I found that very interesting as well. And some isolates that aren't highlighted are either the ladders or they're from the isolates that didn't key out as Vibrio cholera. So this is the phylogenetic tree that was built. With the blue arrow, this is the ornamental Vibrio cholera that seem to be the primary pathogens. As you can see, they form a clonal group, and they almost had 100% identical sequences across the 20 different isolates. And then in green, these are the catfish isolates, which were more diverse, having multiple genetic variants, leading us to believe that the catfish Vibrio cholera could be more opportunistic. And then In red, those are 010139 Vibrio cholera, which are from human outbreaks in Nepal, Brazil. And these isolates also formed a colonial group, as well as they formed their own clusters separate from the ornamental and the catfish isolates. So this is a histopathology, and Dr. Camus conducted all the histopathology, and I'm just reporting his findings. So this is an image of the intestine, and you can see damage to the epithelial lining with the hemorrhaging, and bacterial rods are also seen in the slomic cavity, which is off to your left here, represented in the asterisk. And then there's also very little inflammation, and this leads us to believe that it could indicate a quick death, so damage occurs very quickly or acutely. And then there's also acute necrosis in the intestines, which is also shown by the arrows, which is the pale pink in the image. So this is an image of the spleen, and it is enlarged, and necrosis, which is also seen by the pale pink images, are surrounded by macrophages, which could indicate an immune response, which could also explain why it was enlarged. And the damage could be from the toxins and not the pathogen itself. And this last image is just to show you the comma-shaped bacteria that is there, which is suggested of the Vibrio species. And the presence of the bacteria in the gills also indicates a generalized septicemia. So for my disc diffusion, the isolates were inoculated on Mueller-Hinton blood agar plates, and the antimicrobial discs used were fluorophenocal, oxytetracycline, and sulfadimethoxine, and then the zones of inhibition were measured. And for the BBO crystal kit analysis, the isolates were suspended in BBO crystal inoculum fluid, and the profile numbers were calculated and entered into the BBO electronic codebook. So for my results for the disc diffusion, all catfish isolates were sensitive to all 3 antimicrobials, and 10 non-catfish isolates were non-responsive to oxytetracycline, and 2 were non-responsive to sulfadimethoxine. So in the image underneath the disc diffusion, that plate in the middle is a catfish isolate, and as you can see with the zones of inhibition, they were sensitive to all 3 antimicrobials. And then the 2 plates alongside it, I just wanted to show you what it looked like when it was non-responsive. You had growth all the way up to the disc. And for my BBL crystal analysis, very few isolates correlated with the appropriate code that we found for the sequencing when we used those primer sets earlier. And this could be because there really isn't enough information in the BBL database to get consistent identification results through the analysis. So in conclusion, for my sequencing, each primer set demonstrated high variability against all 66 isolates. Gyrase B was the most successful, and 6 different Vibrio species were identified, the most frequent being the multiple strains of Vibrio cholera, Vibrio mimicus, and Vibrio vulnificus. And for my cholera toxin screen results, catfish isolates were PCR negative for the CTX-AB gene, and they also clustered separate from the 010139 strains in that phylogenetic tree that I showed, which suggests that they are non-toxigenic, non-010139 strains. And for my disc diffusion, all the catfish isolates were sensitive to all 3 antimicrobials. And for the BBL crystal kit analysis, like I mentioned, there really wasn't consistent results that could be used due to lack of information in the database. And for my RET PCR, DNA fingerprinting and gene sequencing suggests that catfish and ornamental fish Vibrio cholera isolates can represent different genetic variants. And for the phylogenetic tree, the isolates from catfish were highly variable, while isolates from the ornamental fish formed a clonal population. And this would suggest that isolates affecting ornamental isolates are more likely to be a primary pathogen, while the isolates from catfish are more likely to be opportunistic, infecting due to stress from the overcrowding or the high temperatures and in the hatcheries. So applications for future research, the CTX screen can be used in order to assess the risks associated with infection from the working in the hatcheries. And differentiating the Vibrio subspecies allows for easier, more reliable identification along with future treatment options. And understanding and characterizing the Vibrio species could aid in prevention and detection for future outbreaks in hatcheries or commercial ponds. And we also sequence the genomes, and this can be used to study further comparative phylogenomic studies to better illuminate the relationships of the fish Vibrio collar isolates to other isolates associated with human health and aquatic animal health. So I'd like to give out a thank you to Dr. Matt Griffin for supervising and giving me the opportunity to complete this project. And I sincerely appreciate the opportunity received from Mississippi State for allowing me to use their facilities. And I'd like to thank Dr. Stillwell, Dr. Petty, Dr. John Hawk, Dr. Soto, and Dr. Walsick for sharing their isolates. And I'd also like to thank Miss Cindy Ware for her help in the lab and Dr. Camus for his help with the histopathology. And that's it.
All right, thank you very much, Allison. That's an impressive project for your time as an undergrad. If there's any questions, feel free to unmute yourself, or you can put them in the chat box and I can read them out. All right, any questions out there? All right, Allison, you got a question here in the chat from Patty. Was the possible toxin seen in the ornamental fishes since there was no toxin production genes in the catfish isolates?
So it isn't, we didn't have like consistent, there was a lot of non-random banding when we ran that toxin screen. So like you said, in those ornamental fish, we also had the isolates that contained that Vibrio vulnificus. When we ran the screen initially, they did look like they had a positive band, but it's like I said, it could be from non-random banding. I'm not really sure what those could be since they did have a lot of messy results. So I really don't know for that, but any isolate that was identified as Vibrio collar, like at the beginning, I can go back to the image. Sorry. Okay, so these first 1 through 20 were the ornamental catfish. As you can see here in the screen, there are some included that had consistent results that were negative for producing those CTX-AB genes. But like I mentioned, the ones that are excluded, yeah, there's at the moment, we'd have to look into more finding a better way in looking and trying to identify if they have those genes.
All right, thank you. We have another question from John Drennan. He's wondering if the next step is to screen the catfish isolates for sensitivity of antibiotics typical of human health.
Yeah, that actually would be extremely interesting to see. And at the moment, those antibiotics that all the catfish isolates were susceptible to them, and that would be really interesting to see how they would work for the human health.
All right, thank you. And Esteban, I see your hand up.
Hi, Allison. Great job. I was curious. I know that Dr. Griffin has a lot of issues washing his hand after he goes to the restroom and then go into the catfish ponds. So I was just wondering if you guys have considered reaching out to the local hospitals, and just see if there is a bank of potential Vibrio, isolates, because what you guys are calling Vibrio cat isolates, from catfish might actually be some sort of like, reverse seeding of the ponds. So I was just curious, if there is any, local folks, that you can reach out, and see if there's a collection of Vibrios, from human sources, that you can compare, because you guys have now a very nice collection of isolates that could be interesting to see that comparison.
Yeah, that would be real interesting because like I mentioned before, the isolates that we have now for the outbreaks are not local. They're from outbreaks in Nepal and India, and including Brazil as well. So yeah, that could be another thing to look at as well, because we didn't have any local Vibrio cholera isolates that we included in that.
All right, thank you. And Jim Winton, I see your hand up.
Yeah, thanks, Allison. That was interesting. One thing that I wanted to know was most of us think of Vibrios as brackish or marine organisms, and here I was a little curious to see these being so pathogenic in a freshwater environment. Did you do any saline enhancement to look at growth and also at toxin production? Rita Caldwell showed that toxin production increased with salinity.
Yeah, no, we did not. We only, from what I did, we got the isolates from other associated laboratories, and we didn't look at any increased saline. We just initially put them on to the TCBS auger and went from there. So that would be really interesting as well.
All right, thanks.
Any other questions? Okay, well, thank you very much, Allison. Excellent presentation and good work, and get a lot of compliments here in the chat and virtual round of applause.
All right, thank you.